just got an aliquot of a plasmid from my colleague, and the first thing I'm going to do is I'm going to transform it into DH5 alpha and sequence it. So I'm the DH5 alpha dog of the lab and don't have to worry about there being mutations I didn't know about. That's a really important thing to do whenever you get a plasmid, is to sequence it. And in order to get more of the plasmid so that you can sequence it, you can stick it into cloning cells like DH5 alpha, get the cells to make lots and lots of copies of it, and then send those copies for sequencing. And nowadays you can do like whole plasmid sequencing instead of just focused one, and you get the sequence of the whole entire thing. And sometimes you can't even get the sequence when you order a plasmid. It's really important to have the sequence so that you can then do cloning with your clone, and so that you don't have any extra or changes in the thing, and then you think you're trying to make a protein and then it's not making the right protein, or it's not making the protein, and you don't want that to happen. So let's talk a little bit more about this.
Molecular cloning is really awesome because we can stick the instructions for making something like a protein into a circular piece of DNA called a plasmid, and then stick that into bacteria and get the bacteria to make lots of copies of it. And if we put in the instructions for making a protein, then we can also get the bacteria to make the protein for us. But we use different cells in order to do this. And so we use like a cloning cell when we just want the bacteria to make copies of a plasmid, and then we can stick it into expression cells to actually get them to make the plasmid. And we can do this because different strains of bacteria are optimized for doing different things. So for example, we talked about how we can use these strains of BL21 that are really good at making proteins on demand. So they have this inducible system that I'm not going to get into here that allows you to add IPTG to make them make T7 RNA polymerase, which then if your gene is under the control of T7 RNA polymerase promoter, your protein will get made. These BL21 are also good for making proteins because they have mutations that would knock out proteases that could chew up your protein, as well as mutations in both the restriction and methylation system to keep the cells from chewing up your plasmid. There are also other bells and whistles, like cells with rare codons and that sort of thing. So these cells are really, really good for making proteins. But what if what you care about more is you just want to get them to make a lot of copies of the plasmid for you? There are multiple reasons why you might want to do this. One is just to propagate the plasmid so that you have a stock for it going forever. Another reason is if you want to use the plasmid for something like in vitro transcription. So you're using it for the making RNA from it and not making protein from it. So you just want a lot of that plasmid. Another reason is maybe you are going to then subclone it into a sort like a BACMID or something, so that you could do insect cell expression or expression in some other sort of cells. So there are reasons why you would want to put your put a plasmid into bacteria that are good at making copies of the plasmid and keeping the plasmid around and not chewing it up, but you wouldn't want them to be making the protein from it. Your protein might even be toxic to the cells. And so enter the cloning cells. And so a lot of the cloning cells that we use are of this DH5 alpha strain, which is a derivative of this K12 strain. And it has some key mutations that make it really good for cloning. And we'll get into some of these mutations in a minute. But I just wanted to get back to what I started this post on, is that a common time that we put a plasmid into these cells is if we want to actually isolate the plasmid. So we take the plasmid and then we put it in these cells so that the cells will make a lot of copies of the plasmid. And then we can then isolate, purify out those plasmids using a method called a mini prep or an alkaline lysis. And then we can either use it or we can sequence it. And it's really important that we sequence plasmids whenever we get them because there can be mutations. So bacteria are really great in that they'll make lots and lots of copies of this plasmid for us. So we can use PCR to do things like make copies of a region of the plasmid, but the bacteria, all we have to do is give them this plasmid and they'll make lots of copies of it. There are even high copy number plasmids where basically we can get the cells to stock up on a lot of copies of the plasmid within the single cell. And so the plasmid itself can control too how many copies of this you have in there based on the promoter and stuff, which I go into in a different post. But the bacteria, they make a lot of copies. And so even if they're not making a lot of copies within a single cell, these cells are going to be doubling frequently. And so you're going to get lots and lots of bacteria making lots and lots of copies, and something can go wrong. And so you can get mutations in the sequence. Sometimes you can get mutations in the backbone, other times mutations in your actual sequence. Sometimes these don't really matter, but sometimes they really do matter. And sometimes it can be the reason why your protein isn't expressing is it turns out there's a mutation that you didn't know about. Or the reason why your cloning isn't working is because there's a mutation that you didn't know about that's making it so that your primer doesn't bind or that your restriction enzyme doesn't cut. So it's really important that you sequence your plasmid to make sure that you know that everything is okay. And also, even if you purchase a plasmid from like a repository, like AdGene or DNASU, or maybe you actually buy a plasmid from a company, they're often, especially for the companies, they don't give you the actual like sequence of the plasmid. And so you want to go ahead and sequence it yourself especially if you want to do any sort of cloning that's going to require you to design primers or design that match the sequence, or to design restriction cut sites, and that make sure there aren't extra cut sites where you don't want them, that sort of thing. So it's really important to have a sequence. And what's really great is that nowadays we can actually get this whole the whole plasmid sequenced thanks to long read technologies, things like PacBio and nanopore technologies that give you a lot longer reads, so you can sequence the whole plasmid as opposed to just being able to sequence a shorter piece of it, such as with Sanger sequencing, where you also have to like provide them the primers. So then you have like two reactions if you want to go one side on each insert, and then you're only getting part of the plasmid, and you end up paying the same as what you're paying for a whole plasmid nowadays. So we in the lab I just joined, we use like apparently use Primordium. I've never used them, but I'm going to try them out hopefully in a couple of days, assuming that my transformation worked. So in order to get the plasmid to sequence, we have to stick it into the cells, and this is a process called transformation. Then we use antibiotic selection. So we grow them on a plate that has an antibiotic that matches an antibiotic resistance gene our plasmid has. So only our plasmid will be able to grow. Then we can, when we plate this, each of the bacteria that take in the plasmid and then take hold on the plate, they'll start doubling on top of themselves, and you'll get these gloopy things called colonies. Then what I'll do is I'll pick a colony and I'll do like a 5 mil growth, and then grow it overnight. And then I can do a mini prep and purify it out. So normally if I was doing cloning or something, I would have, I might do some sort of pre-check, like a colony PCR or an analytical digest, to see if the cloning likely worked. This won't tell me for sure, but I can get an idea. And I've talked about this more in other posts. But when you're starting with a purified plasmid, so when you're starting with like a mini prep plasmid or some plasmid that you buy, here it's a lot easier for the cells to take it in. And it's a lot, so you don't have to transform as much. I just transform like a single microliter typically of this. And then I'm not going to plate as much, because if I was to plate a lot, it would overgrow. Whereas when you're doing some sort of cloning, especially if it's a cloning method that requires the bacteria to do some work, then you're going to to stitch together things like a ligation independent method or that sort of thing, then you want to make sure that you're plating a lot. And so that typically then we like spin down the reaction, we resuspend it and do that sort of thing. But so that's just something I want to note, was that if you're going straight from a mini prep, then you can transform less, plate less, and then go straight to this mini prep and sequencing. Okay, so that is what's on tap for me, assuming, so I did this part today and then I plated it, and then hopefully tomorrow I'll be able to do the overnight growth to get enough to do the mini prep. And I have more on all those posts and other, those things in other posts. But why I got into this was because I transformed them into these DH5 alpha cells. And so why did I do that? What makes these cells good? First of all, what is DH5 alpha? So I told you it was this, it's derived from this K12 strain. And so the name DH5 alpha, the initials are for its isolator, Douglas Hanahan. And then the alpha is because it has this alpha complementation allele that's going to allow you to do blue-white screening. So what this means is that in blue-white screening, it's this technique that you can use to see if your insert likely got into your plasmid during the cloning step. So the antibiotic resistance gene is going to tell us whether the plasmid got into the cells, because only the cells that have the plasmid will be able to grow in the presence of the antibiotic. But that doesn't tell you whether your insert got into the plasmid. When you use a blue-white system, what you basically have is there's this thing, this beta galactose, and the bacteria use it to break down lactose. And so I go over this in other posts, but the basic idea is that part of this protein that we can give them this chemical called xgal, and it'll break it down into this blue product if it has functional beta galactosidase. And so in order to make this functional protein, you need the whole protein. And the way that they have it set up in these cells is that part of the protein is going to be present in the bacteria, because the bacteria normally have this protein, but there's going to be a mutation. And then this mutation can be rescued if you provide the region that is missing in these cells on your plasmid. And so then if you interrupt that, then you can knock that out, and then you can use this. You stop making the blue product, and so you can use this to see if your colonies likely have the insert. So these DH5 alpha cells have the presence of that, which allows you to do this blue-white screening, which is really cool. And another cool note, and so apparently Douglas Hanahan, he was actually at Cold Spring Harbor Laboratory where I did my PhD, and then he went to UCSF, which is where I am now doing my postdoc. But now he's currently, I think, at the Swiss Federal Institute of Technology. But cool story how that came. We have similarities in our scientific wanderings. Okay, he also did things like determine ways for transformation, a bunch of other really important stuff that's really important for microbiology and molecular biology techniques. Okay, and so the 5, I don't know, I think the Wikipedia page said it was like the 5th work best, or I don't know, something like that. But anyway, okay, so what else is good about it? It has the ability to take in the plasmid well, so it's got a high transformation efficiency. So transformation is where we get the cells to take in the plasmid. A common way that we do this with bacterial cells is we use these like chemically competent cells. So we can weaken the cells, the membrane, and then get them to take in this plasmid when we heat them up, and kind of it opens up these little pores, and then we have this calcium that's going to help it get close, and it sucks it in. These DH5 alpha are good at this for a couple of reasons. One of them is that they have this REL-A1 mutation that's going to alter the membrane composition, so it's easier to get into. And it but that's not all it does. It also like relaxes the cell, so it like removes it relaxes its response in regards to when it's starved of like amino acids. So the protein building blocks. Normally, bacteria would halt their production of RNA. but this mutation allows them to keep transcribing RNA even if they can't make protein from it yet. So that isn't really involved with the transformation or that sort of thing, but this is just one of the mutations that this strain happens to have. And so it's not just that it allows for letting things in, there can be other effects. And so here's an article, I'll link to some articles. Another thing is that it has this mutation in this like DOR mutation, which apparently makes it transform more efficiently. And so I will put a link to this page. It says that it seems to assist in the uptake of larger DNA molecules. DOR is a transcriptional regulatory with DNA binding activity that controls expression of a set of genes involved in deoxyribonucleoside catabolism. So catabolism is breaking things down. Deoxyribonucleoside is DNA. So I'm guessing that this mutation prevents it from chewing up the DNA and somehow helps with the transformation. But I'm not exactly sure, and I'm not going to try to speculate. Okay. But so now your plasmids got in there, and now we want it to not mess with our DNA. So we don't want it to chew it up. So there's this REC-A1. There's this REC-A protein, which is needed for some types of homologous recombination. So this is a method where kind of the bacteria will find regions that look, if there's like a damage to one DNA, it'll like try to find the part that matches and then fix them, and then but if it like fixes them wrong, it can end up shuffling things around. And so if we don't want our things shuffled around, especially doing this cloning part where we're trying to get these really specific sequences exactly the way we want it, we've designed this plasmid, we put it in there, and we don't want to change things around. And so this REC-A mutation helps with that. Also note that they have other forms of recombination machinery that are still intact, and this is going to allow us to do ligation independent methods of cloning, where basically we rely on homologous recombination in the bacteria to fix to like piece together DNA pieces that we put in there to like stitch them together and stuff. Okay, there's also a mutation in this END-A1, which is an endonuclease 1, which is this non-specific DNA chewer. So this mutation prevents it from chewing up the DNA. Okay, so this weird thing, this is telling you about that there's a mutation in its restriction enzyme system, but not its methylation enzyme system. So this like restriction methylation system is this thing that bacteria use in order to protect their own DNA, but destroy foreign DNA. So they modify their own DNA with this methylation marks, and then they can cut up any unmethylated DNA using a restriction enzyme. So this methylation is going to like hide those restriction enzyme cut sites, but then this methyltransferase can if the methyl marks aren't there, such as if you put foreign DNA in there, then they can get chewed up. So if we're going to stick a plasmid in there and our plasmid isn't methylated, then this cell can chew it up, except that it has a mutation in this restriction site, its restriction system, so that it won't chew up your cells. And so just to note that, so here we have this RK minus MK plus. When we're looking at the BL21, this actually had a mutation in both of these, and it had this DCM mutation. And so this is another methyltransferase that it was mutated in. So when it comes to actually the sequence, the methylation, one of these important methyl markers, these enzyme that's going to, the STAM methyltransferase, what it's going to do is it's responsible for kind of methylating the bacteria's DNA when the bacteria is like making copies of it. And so this STAM is going to methylate things, and this is going to be helpful because it's going to let the bacteria recognize which strand is the new strand, and it's also going to let it protect its DNA. So an important thing about these DH5 alpha cells is that they don't mess with this. They don't mess with this damn methyltransferase. And why this is important is because it's going to let you do things like and why this is important is because it's going to let you selectively degrade the parent's plasmid when you're doing a PCR-based cloning method. So with methods like SLIC, where we're using a template or using a plasmid as a template for making copies of things that we then stick together, or making copies of like the whole thing and various things. But anyway, when we use PCR to make these copies, then we're doing this in vitro, we're doing this in the test tube, we're doing this outside, there's no methyltransferase there. And so it's not going to get methylated. And so we can use this, we can add the restriction enzyme that's going to cut it up, anything that is methylated. And so there's this weird restriction enzyme called DPN1, and it's going to recognize methylated DNA. So your parent plasmids are going to be methylated, but the PCR products aren't. And so then you can degrade the parent, which is important so that you don't have to worry about the your parent plasmid being what's actually getting into these bacteria, because it's going to have an easier time doing so because it's all it's already all whole, and it doesn't have to be stitched together by the bacteria, and so it has the antibiotic resistance gene, so it would be selected for, and so yeah, so this DPN digest is a really important tool, and we can do this with these strains because they still have the ability to do the methylation. Okay, and then as we talked about, they have that mutation in their in the cells that has basically, it has the beta peptide part that complements the alpha peptide that's provided by the plasmid in the blue-white screening. And so you can look for the presence. Okay, a couple other things. There's a couple other mutations too. So it has mutations that are like different from the parent strain or different from other strains, but not necessarily relevant for our purposes. The cells just happen to have them. So there's like this JARA96, which gives it nalidixic acid resistance. Thi1 makes it a thiamine oxytroph, so it can't make its own thiamine. It has this other mutation that leads to increased hydrogen peroxide resistance, and there's some others too. And yeah, just a little bit more about this DH5 alpha. So as I mentioned, it's from this like K12 strain. This is like the wild strain. It was isolated from a patient's stool in 1922 at Stanford. And basically this was the original strain, but then bacteria, as I talked about, they like to mutate. And scientists actually can speed up the mutation with radiation or adding chemicals called mutagens that are gonna like make breaks in the DNA or keep the bacteria's fixing machinery from working. And scientists have done this a lot to this K12 strain because it lacks virulence genes, so it can't hurt us. So it's considered like non-pathogenic. And so you can take this K12 and you can mutate it in various ways to get other important strains. And so K12, mutate leads to MG1655. And this is going to mutate, give rise to DH5 alpha and its cousin DH10B, or top 10 if for some of the commercial strains are called that. And so these DH5 alpha and the DH10 are both these K12 strains, as are some of the other ones that I've talked about, like origami strains. I think some of like Nova Blue. Yeah. And just it's a single circular chromosome, about 4.6 million DNA letters, and about 4,500 genes. And it's super duper helpful. And so hope that helps you understand these DH5 alpha cells.